Opportunity to address such a distinguished audience. It's a great pleasure for me to introduce Dr. Zeeuw. He has a PhD in neuroscience from the Erasmus University of Rotterdam and an MD from the Erasmus University of Rotterdam as well. Additionally, Dr. Zeeuw is the founder of PPI Lab and currently works there as an advisor. He also works as a director of Neurasmus, BV. He was the president of Neuropathic Pharmaceuticals, PET, in short, in the Netherlands. So far, Dr. Zeeuw has published several scientific articles which have been internationally recognized as contributions to the field of behavior and neuroscience. Please welcome Dr. Zeeuw.
Damos la bienvenida y el agradecimiento entonces al Dr. Zeeuw y les dejamos con su conferencia. Que lo aprovechen.
Gracias mil centos. It's really a great privilege for me to be here. I've been in Ecuador several times, and some of my PC, Fellows, has been working here, and ever since the connection has been very tight. And I will also show you data in my talk that show proof of principle of the strong collaboration between Rotterdam and Riobamba. Now, what you see here is, I think, a very, very special mountain, not just for Riobamba, not just for Ecuador, but for the whole world. This is the mountain, Chimborazo, yeah, that is closest to the stars, if you think about it. So, I always thought, of course, if you're on the top of the mountain, on the peak of the Mount Everest, you're close to the stars, but this is not really true, because this mountain is so close to the equator, and you probably all know this, and since the Earth is wider than long, you are actually on the top of the Chimborazo, closer to the stars than when you're on the peak of the Mount Everest. So this is a very special place. And I've noticed this before. The previous time I was here, I actually have been running up the Chimborazo, which I did regret at some point, but because I couldn't get the oxygen to let's say, recuperate in a quick instant. So it took a few minutes. But I have a special feeling for this mountain, and that's what my talk is all about. So I will try to explain to you what's going on in your brain when you try to climb onto the Chimborazo. What's going on when different brain regions work together, and particularly what's going on in our small brain, in the cerebellum. So, next, yes. Here you see the position of the cerebellum, the small brain, in our entire brain. And what you see is that it actually controls, through the cerebellum nuclei, via the thalamus, the cerebral cortex. So the cerebral cortex is our big brain. Yeah, and this interaction between the small brain and the big brain, I think, is very critical for many functions, including those what you need when you're climbing on the Chimborazo. So, usually when you think about, okay, I'm going to climb on the Chimborazo, you make this decision in your prefrontal cortex, here in the cerebral cortex, and then you mimic, you send the signals through various pathways, through your motor nuclei, so that you can move your limbs. At the same time, you have sensory feedback, and you try to do that. But if you don't have a cerebellum, you will basically walk like a drunken guy. So you will make movements too early or too late. You will have movements that are reaching too far but too little. So you look like a drunken guy. And to make this timing perfect, you need the cerebellum. And the cerebellum works like this. It projects onto the nuclei and then controls through the thalamus and the medial landscape junction and the pontine nuclei. It controls this process, what the cerebral cortex is doing ultimately with the motor output. Yeah, and how that exactly works, I'm going to try to explain in this talk. And what is really critical is that if you look at the neuroanatomy of the cerebellum, is that you see there are these stripes, what we call sagittal stripes. And each and every stripe, here you see it colored with a marker, zebrin 2. But each and every stripe is involved in particular movement. So in the center, for example, you control the trunk muscles. So when you walk up the Chimborazo, of course you want to keep balance. Well, that's controlled right here. Yeah, the blue and white piece. Then if you go more lateral, you will start to use your arms. If you go even further, you will start to use your fingers if you want to touch a rock. Yeah? You also need eye movement control, controlled by the flocculus. I will talk about that as well. The most fundamental basic balance principles are controlled here in lobule 10 and lobule 9, the modulus and the uvula. So all of those regions are important for motor control. And what we're finding out lately, and I will not talk about that, but it is still worthwhile mentioning, is that even if you go more to the lateral side, you will even control cognitive processes that Mrs. Bilyashvili will talk about in the next talk. So the cerebellum is really controlling all sorts of aspects in sensory motor control as well as cognitive control. Here you see, if you, for example, image the activity of the fingers. So when you climb with your fingers on a rock of the chimborazo, you want to pull up yourself. Well, even to the level of the small fingers, there's very special microzones in the cerebellum. This is the whole cerebellum of the human, that can be lighting up when you move these specific fingers. Yeah, so it goes to a very high level of detail. Here you see, again, the cerebellum, but now of a mouse, labeled with Zeppelin 2. So you see all those sagittal zones. Some of them are very dark, like the ones in the flocculus that control the eye movement here on the side, but others are lightly stained, like in the anterior lobe. And what they really do and how this really works is very important for understanding how the cerebellum can operate all its functions. What I, in the end, will try to convince you of is that because of all these different protein expressions in these different zones, there's intrinsic properties within the sagittal zones that allow the system to adjust to what is needed. For example, if you're in the chimborazo, the oxygen level will be lower, right? So you need to have a different interaction between your limb muscles and the way you breathe. Well, all of that is controlled by the cerebellum. Here you see that these zones in the cerebellum, this is again the cerebellum of a mouse, yeah? They are not only different because of that one has Zeppelin 2, which is aldehyde C, and the other one has not. There's a whole series of genes. So nowadays, with modern technologies, so-called slide set technology, you can, in a single section, you can determine the proteins of hundreds, if not thousands of proteins at the same time. And then you can do some clustering analysis, and you can see that here in this upper quarter, yeah, all the genes that are associated with aldehyde C genes. So it's not just one gene, it's a whole series of genes that control the functions, that determine the intrinsic functions of the aldehyde C positive zones, which is here colored in light blue. And then the other zones that are negative for aldehyde C, they're actually all positive for PLC beta 4, and there's other genes associated with them. So again, there's really intrinsic biochemical pathways that determine the optimal functions of different brain regions, in this case of specific zones in the cerebellum. This also comes with differences in electrophysiology as well as in connectivity. So if you look, for example, here at the top, you see the recordings in a red zone, which is the Zeppelin negative zone, and in the Zeppelin positive zone, which is a blue zone. Yeah? And what you see is that the firing frequency of what we call simple spikes, those are the deflections that go down, yeah, they are higher in the Zeppelin negative zone, so in the red zone. And so they are lower in the Zeppelin positive zone. On top of that, you see so-called complex spikes. The complex spikes, whenever there's a complex spike, which is generated by the climbing fibers that move into the cerebellum, then there's a period of silence of simple spikes. So it means that if the period is shorter, that the simple spike firing frequency must be higher. So this is again true for the Zeppelin positive zone, or negative zones. And here you see the opposite for the Zeppelin positive zone. The same thing is true for the connectivity. It's subtle but important, and I will show you in a clinical example later in my talk why this small difference is clinically relevant. So here you see a blue zone, yeah, which is the Zeppelin positive zone, and all the connectivities. The final output comes from Purkinje cells, they go to the cerebellum nuclei, and then they go into the brainstem determining the outputs, they influence the cerebral cortex, and motor neurons downstream, etc. They get the input mostly from granule cells, which give off parallel fibers that traverse multiple modules. So it's not that only the blue module gets input from blue granule cells, no, actually, blue module gets input from all parallel fibers, also from the Zeppelin negative zones. But there's one big difference, it may seem small, but this is the unipolar brush shell, the UBC indicated here. They are there in Zerebellum positive zones, but they are not there in Zerebellum negative zones. And if you put all these properties together, when we discovered that there was a difference in firing frequency between the different zones, we started to make hypotheses how the learning takes place. And the idea is that because the firing frequency is lower in the blue zone, that there's more room to increase the firing frequency there during the learning. And that in the red zone, it's the opposite. There, the firing frequencies are really high to begin with, like 100 Hertz. And there's ample room to bring it down. And I will try to convince you that this is actually the case. So therefore, we start to call one upbound, the blue zones, and the other one the downbound zone, the red. When we tried to prove this, this was in the paper with the Chinese guy, Zeeuw, which he published in eLife. We just randomly screened the cerebellum of mice, and we went from, let's say, Zerebellum negative zones to Zerebellum positive zones to Zerebellum negative zones. You see it here too. So these are Zerebellum positive cells. You can see this in vivo because you can see the green fluorescent protein lighting up under the microscope. And then you go in with an electrode and you can identify the spike frequency. So these are the simple spikes, which can then be low, relatively low in the blue zones and can be high in the red zone. And so we did this and we went from anterior to posterior, from left to right, and we found that this was a very consistent difference. So we came up with the following hypothesis. As I said, so the baseline firing frequency is low in the blue zone, but then there's ample room to go up. It's high in the red zone, then there's ample room to go down. So we call it upbound, because it goes up during learning, and downbound modules. And this comes together with other activities of the complex spikes and molecular interneurons that I will come back to later. But the critical point is these changes in the simple spikes, here revealed at the top. It's the opposite. There you want to decrease the activity of the parallel fiber to Purkinje cell synapse, so it's long-term depression, and you want to increase the effect of the molecular interneurons. So there is long-term potentiation here, long-term potentiation there. When you do all of that, simple spike will go down, and interestingly, all these forms of synaptic plasticity are driven by an increase in climbing fiber activity that you see here. So these are the basic hypotheses that I will try to prove to you. If I can have the next slide. So these are the two main parts of my talk. I will first now give you a series of experiments in which these hypotheses are, let's say, not proven for 100%, but made very likely. And I will end it with a clinical example. And then in the second part of my talk, I will talk about sensory motor learning in a downbound module, where I do the same thing. I show the basic principles, I show the evidence, and I end up with some clinical studies that we're currently doing, actually together with people here on Rio Mama. So, next slide please. So we start with the upbound module. In the upbound module, you can see there's all these Zerebellum positive zones indicated in dark gray. I will focus on one type of behavior controlled by the flocculus here, because it's easy, it's completely black, as you see, so it's completely Zerebellum positive. And the behavior that's controlled by timing behavior can be readily tested, because you can test the inputs, you can test the outputs, and you can record the neuronal activity in between very accurately. So let's go to the next slide. Yes, maybe you can now, again, click one more time. Yes, let's stop it like this, yes. So here you can see how strong these reflexes are. Look at the stability of the eyes and the head of the owl. No matter what the experimenter is doing, even when you cover the eyes, actually the head is still stabilized, right? So it's a very strong vestibular reflex, both for the eyes and the neck. And we can mimic this in the lab. Next slide, please. Sort of fool the animal by giving a different visual feedback that he normally has when he moves his head in space in real life. Now we give more retinal slip because there's a visual image moving in the other direction than the tables. And this will induce an increase in the strength of the eye movements. If you do this for like half an hour and then put the animal back in the dark, you will see that the amplitude of the eye movement is increased. Next slide. Here you see the pathway at the bottom that's responsible. So here you have the Purkinje cells and the flocculus. In the end, it controls, through the floccular target neurons, the eye muscles, the abducens nucleus neurons, and they control the eye muscles, right? But they're driven here by vestibular input. And this whole plasticity that is occurring here at the level of the Purkinje cells is driving an increase in the eye movement in the end here. And so the test that we want to do, because the hypothesis is that in the upbound microzone, the simple spikes will go up. Yeah, so what we did is we subjected the animals on this turntable. We give this outer phase stimulation of the visual world with respect to the head movement, with respect to the vestibular stimulation. And then we start to record the eye movement here in the flocculus on the left side. And then if you click one more time, then you will see here on the right what the activity looks like. Here you see it, right? So you stimulate, you increase during the learning the amplitude of the eye movements. Here the amplitude goes up, and at the same time you see here with the probe recordings that in the same probe trials the activity of the simple spikes will go up. Here you see it quantified. So this at least means, it doesn't directly show that it is causal, but at least there's a very strong correlation between the activity of the simple spikes in the Purkinje cells in an upbound direction when the amplitude of the eye movements go up. Yeah, so this is a strong evidence, yet it doesn't prove it. To prove it, what you need to do, you do not only need to show that there is this correlation, but you should also show that it is necessary and sufficient. So therefore we did the next experiment. Next slide, please. We started to use optogenetics. Optogenetics, many of you probably know, is a wonderful technology. You can bring in proteins in a cell-specific way, so in this case specifically in Purkinje cells, that only then expresses proteins, and this protein is sensitive for light, for a certain wavelength, in this case 470 nanometer light. So we bring this in a genetic way, or with viruses, you can do it in multiple ways. And then you bring the mouse back on the turntable, and you start to stimulate the light. And if the hypothesis is true, if an increase in simple spike activity is sufficient to drive the eye movement, then you should see when you crank up the activity of the Purkinje cells, now not by stimulating with the table, but just giving a light stimulation in different intensities, you should see accordingly an increase in the eye movement amplitude. And this is indeed what you see. So here I show two different intensities. Yeah. When you crank up the light, there's more simple spike activity, and the eye movement amplitude is increasing accordingly. So this proves that an increase in simple spike activity is sufficient to drive the eye movement. Next slide, please. To show that, which is another part of the hypothesis, right, that this occurs through potentiation of the parallel fiber to Purkinje cell synapse, here the L2P, we had to corrupt or disrupt the specific process of long-term potentiation at this synapse. And this is what we did in the next slide. We took care of the presence of the GLU-A3 AMPA receptor subunit. This is a very special receptor subunit. It turned out that they form dimers with GLU-A2, and then when they are activated, the diameter of these AMPA receptors is actually increased. And so the ionic flow through these receptors is increased. And this is exactly what happened during the learning. And if you take out GLU-A3, you can see that the learning that normally takes place does not take place anymore, or to a much smaller extent. So this too confirmed that the LTP process is critical to crank up the simple spike activity. Next slide, please. Now, I showed you in the beginning of the talk that there's these UBCs, the unipolar brush cells. And I will show you in this video that they are actually also important for diseases and even for the occurrence of ataxia. Please, can you click one more time? Here you see a patient, Mrs. Dijkhuizen. She in the meantime passed away, but this is an old video of 15 years ago. And there you see the Professor Silke Smith from neurology. he had this patient suddenly appearing with very strong ataxia, and he couldn't find out what was going on. So he knew I was interested in the cerebellum, so he asked me, how can we look at this? And what we did is we asked for a bit of serum from the blood from this patient. Right? You can clearly see she's ataxic, right? She cannot stand by herself. And so we asked for the blood, the serum, and we put the serum of this patient just in sections of a rat and a mouse. Next slide, please. And here you see what we saw. We saw this huge number of unipolar brush cells labeled in the upbound microzones. Yeah? And we recognized this pattern because there was a combination of specific labeling in the hippocampus and other regions, and we felt, well, this could actually be the distribution of mGluR1, which is typically distributed in these unipolar brush cells. And just by the neuroanatomical image, which appeared unique to us, even though there are like 30,000 genes, we thought this might be mGluR1. And so, in the next slide, we tested this hypothesis. And here you see again the patient serum on a wild-type section of a mouse. Yeah, you see there's a lot of labeling in the cerebellum, as well as in the hippocampus and other regions. And this made us think, okay, this might be mGluR1. So then we took a section of a knockout of mGluR1. So this mouse does not express mGluR1, and we use again the same serum. And see here it's completely empty. So this really strengthened our hypothesis, almost proved it, that actually this patient somehow had generated mGluR1 antibodies in her body. And this, we found out later, is related to some Hodgkin tumor. And the treatment then was very simple. The only thing we had to do is take out the antibodies against mGluR1, and we did that, and she completely cured, at least initially. And she had also had, of course, still the tumor, so later on she died because of that, but at least the ataxia disappeared when we took out the antibodies. And the strength of this antibody is very apparent in the next slide. You can see that if you apply it within minutes, the biofrequency is changing of the Purkinje cells due to the changes in activity of the UPCs. Next slide. And again, please, one more click, and you will see this mouse walking. One more click, please. Here. Yes. This is a mouse that has only saline. So this mouse did not get the antibody of the patient. But in the other mouse here, please one more click, you can see this mouse actually received the patient's antiserum. So in an acute way, within minutes, when we give the patient serum, this mouse became ataxic. Yeah? And so this showed to us, and when we purified the antibody for only against anti-mGluR1, the same thing happened. So it shows that that the brush cells can be very important, and that diseases purely affecting the upbound microzone can yield these very strong results and phenotypes in the clinic. Okay, so far the upbound microzone. Now we go to the downbound. Here you see one more time that was a recovery. So when we took away the serum, then was a recovery, right? So it was a reversible effect in the mouse at least. Next slide, please. So now we go to the downbound modules. In the downbound modules, as you know, there is no LDLC or extremely little. There's a lot of VLC beta 4 and all the molecular pathways involved. And the best way to test the function of the downbound module is actually IV conditioning. Next slide. IV conditioning does not take place here in the flocus because that's serum positive, but it takes place predominantly in lobule simplex, here indicated in this red box. So the areas that are light, that are serum negative, yeah, they are the downbound microzones. They control what Pavlov described as a conditioning process already more than 120 years ago, for which he got the Nobel Prize. Well, we found that this process of Pavlovian conditioning actually takes place in this region. When you record the activity, you see all sorts of activities related to Pavlovian conditioning. And of course, in our prediction, because it's a downbound module, we predict that their activity should not go up, like what I just showed for the VOR adaptation of the eye movements. Now it should go down. That's why I call it downbound. So let's go to the next slide. This is IV conditioning itself without any neuronal recording yet. So it works like this. You give, for example, conditioned stimulus, which can be a LED light. You hold a small LED light in front of the eye of the mouse. And for like 250 milliseconds, this light is progressing, right? It just stays there, and it co-terminates with the unconditioned stimulus, which is in our case an air pump. And when you repair this, use the next click, you see that initially when you do this on day one, nothing is happening. You get an unconditioned stimulus as soon as you give the air pump, so the eyelid is then closing, as you see here. But if you, one more click please, then over the next few days, you see that the eyelid movements are increasing. Yeah, so if you keep pairing every day for 100 trials or so, the conditioned with the unconditioned stimulus, at the end of day 10, there's a beautiful conditioned reflex in which the eyelid is almost perfectly closed at the moment when the air pump is about to come in. So this is really a very nice way of showing conditioning. Now, so the question is, what's happening with the neuronal activity during this behavior? Next slide, please. We record the activity, as I said, in lobule simplex, so we record again from simple spikes in lobule simplex, and we expect that the simple spike activity will go down, that initially is high, and then will go down to drive the conditioned eye-blink response. Next slide, please. And here you see that this is actually happening, like 100 Hertz, and then it will drop to like 50 Hertz, and it recovers later on. But during the conditioning process, when the conditioned response is taking place, there's a downbound activity. Oh, previous one. What you also see is that actually at the beginning of this stage, there's a little complex spike, just one. But this spike is very critical because maybe you remember I told you that this complex spike is driving all the plasticities in these downbound modules. That you see in the next slide. So here you see when the climate fiber is cranking up, you'll see that there will be LTD, long-term depression, expressed here, a long-term potentiation here and here, so that the interneurons can drive the simple spike suppression. The simple spike will go down because of the increase in complex spikes. To show that this is really true, next slide please. Okay, yeah, next. I show you here a recent paper that just came out, in which we compared the Purkinje cells that are Zeeuw positive and Zeeuw negative. Yeah, so you can see here, this we label again with EA84, this is one of the genes that's positive for LDLA-C, Zeeuw positive, and negative for the downbound microsome. And when we compare the activities and when we try to induce plasticity by stimulating both the parallel fibers and the climate fibers, that's the way to induce LTD, long-term depression, you can see that it's much easier to induce here long-term depression in the red cells, yeah, which are the downbound Purkinje cells, than in the upbound Purkinje cells. So intrinsically, these cells have a stronger propensity to learn, in this direction at least. And some of you may say, well, you know, this may be true, maybe there's different inputs. So we looked also at that. Please, next slide. And here again, you see the different zones where we compare different Purkinje cells and Zeeuw positive and Zeeuw negative cells. And then we stimulated the parallel fibers and we did a test which we call pair pulse facilitation. And you see, this is a way to test the direct input of the parallel fibers without any induction of plasticity, and then you don't see any difference. So it's not the presynaptic process, it is the intrinsic biochemistry of the Purkinje cells that is determining the learning behavior and propensity for downbound learning as opposed to upbound learning. Next slide, please. The other thing that's really necessary to get the activity of the simple spike down, you need to also get this increase in molecular interneurons, because you need to actively bring the activity of the Purkinje cells down by stimulating inhibitory interneurons. And this is also happening during the conditioned response. So here we record from the interneuron, the molecular interneuron here, this cell, and this cell, and you see that that one actually goes up, in contrast to the activity of the Purkinje cells that does go down. Next slide, please. Now, so how can we capitalize on all our knowledge of eye blinking? We have been doing these eye blink tests for 20 years or so, Mafra has been witnessing this with Bas Koekoek, who was the first, and we had Javier Medina coming over, Henk Jan took up on it, we felt them blinking. Many people have been working on air conditioning, and we felt it was time to capitalize. And actually when Henk Jan Boede went to Princeton University, and in the lockdown period of COVID, he could not work with animals because the lab was closed. So he felt this is the right moment to capitalize on this, and this is what we started, the Blink Lab. I will show you what we can do with this also in relation to disease, both in healthy people and in disease people. Next slide. So in the old days, we have already been trying to find phenotype in autistic patients, for example, in our lab. And then we built very complicated sets, headphones, extensive equipment with helmets and puffs, and they don't like it. It's very unpleasant, and it never really worked. It didn't work, but you really had to fight for it. And so Henk Jan developed, together with the whole team, the Blink Lab solution. It's better, faster, cheaper, and more pleasant. And the data we get now with this app, and many of you already here have this app. You can download it from the App Store. You can just put it on your iPhone, and you can do it at home, you can do it with your family, you can do it in the clinic, and you can get wonderful recordings, much better than we ever got in our institutional academic setup. Just because patients are more relaxed, they don't have the terrible puff, you can use a combination of light and sounds as an unconditioned stimulus, and it works really well. And I want to show you some data, especially here, because I have to announce something unique for the people here who are working on Blink Lab. I will get to it. Next slide. So here again you see that we want to do this for neuroscience people working on neuroscience projects, trying to unravel the mechanisms of autism, ADHD, but also other diseases like schizophrenia. And yet we want to use it as biomarkers in the clinic. And of course in the home situation, people can also keep recording and see whether the impact of a certain therapy is working or not. Next. Here you see Stephanie, who's also part of Blink Lab, technician in our lab in Rotterdam. And I want to show you here a result of PPI, pre-pulse inhibition. It works like this. Please, next click. So you should hear a beep. So whenever this stimulus is going on, there's a big beep, and as a result, she will make an eye movement. Now, this is the first baseline measurement. Then what you do is, then you do it again, but with a small pre-pulse. Next one. So now there's a pre-pulse. And next one, please. So small, and then you see that this big response to the big pulse is disappearing, because the pre-pulse is sort of alerting her and saying, okay, don't get too frightened, there's a big pulse coming. And so she can reduce her reflex. It's not a voluntary suppression, it's all involuntary, you cannot limit this, it's all going too fast. But this is what we call pre-pulse inhibition. It works really well in a normal, healthy adult person in our society. Next slide, please. So the question is, what does this look like when you have a child with autism? Next slide. The diagnosis now, as of today, also here, still in Ecuador, but also in the US, of course, is taking a long time. Usually it takes like 4 to 5 years after birth, and you have to do a very complicated test to diagnose autism. And you also get a sort of underrepresentation, because not every country has the same financial means or the same healthcare system. So, for example, the prevalence of autism in the US is 10 times bigger than in Ecuador. But of course, that's probably not reflecting reality. That's just an underdiagnosis, right? Next slide, please. Please click a few times. So normally it's diagnosed here. Another click. But we would like to bring it down with Blink Lab to an early diagnosis, right there, exactly. Because if you can diagnose it earlier, there's a lot of therapeutic possibilities for autism through behavioral training to get those little kids adjusted to a normal society, and they will benefit from it for years. When they're 5 years old, the brain is already a lot less plastic. So we want to develop biomarkers with Blink Lab. And here we show you the recordings with Blink Lab of one of the autistic children that we tested. So again, you see the normal big response to the big pulse. And now we do the pre-pulse. Yes. Again. Again. Yes. And you see now there's no inhibition in this autistic child. So in normal people, you get, when you give the pre-pulse, there's this inhibition. There is hardly any big reaction to the big pulse left. But in many autistic children, there's aberrations with this pre-pulse inhibition. Yeah, and then you can say, well, is this selective to this disease? That's what we're actually trying to prove now. Next slide, please. And we are also, please click a few times. Yes, please, next one. This is the overall view, what I just showed in a nutshell. But you see this huge difference in pre-pulse inhibition effect. Yeah? And you can say it's selective because if you do the same thing in children suffering from ADHD, hyperactive disorder, attention deficit hyperactive disorder, next one. Yeah? You will see, yeah, the next one, that the reaction to the big pulse is the same in ADHD. Yeah? But if you give the pre-pulse only, actually there's a much stronger response than in autism or in the wild type. So also here ADHD has a unique phenotype as compared to autism. The nice thing is, of course, that we all know the use of Ritalin, probably is also used here in Ecuador, for ADHD, not for autism, but for ADHD. And we can even test the impact of the pharmaceutical applications. Next one. Yes. Please click a few times. And then you see the impact of Ritalin within a few hours. So normally in general practice of a psychiatrist or general practitioner, when a doctor gives Ritalin to a patient, it takes usually 4 to 6 weeks to evaluate the impact of Ritalin. Here we do it in a few hours. And we show actually the same kinematics of drug intake. Next one, please. And next one. Yeah, so this is what we do with big data. It's not only ADHD or autism, we now find specific phenotypes, also for selective learning disorders, attention deficit disorders, and so on. So we are very proud of what's going on. We're not there yet. It's really developing very quickly. Next slide. We're doing it worldwide, so now we are reporting as we speak here in all continents in the world. Next one. Please click a few times. You see that all the continents are now recording, even Australia. And I'm very happy to say that actually the greatest recordings, both in terms of quality and in terms of quantity, are being done right here in Riobamba. So I want to thank, next slide please. Next slide. This is Valeria, together with CEO Henk van Boelen. And Valeria is doing a great job, so I would like to give her a big applause. I'm very proud of all the recordings that are streaming into our platform in Rotterdam. Of course, we will publish these things together. The thanks to Valeria is not only coming from Henk Jan, also next slide please. The whole Blink Lab is growing almost every week. Some of them you see here. Yeah? And I would also like to highlight here the role of Maffer. Here she's part also of Blink Lab. She's advising us. She's a scientific advisor just like me. But without Maffer being starting, actually I think in Rotterdam, a long time ago when you started your master, then going back to Riobamba, doing work here, now working in Norway, I think because of all of that, your knowledge in terms of epidemiology, in terms of the mechanisms of the cerebellum, you were really the one that made this possible. And thank you to me. So with this I want to thank also the whole organizing team for this wonderful 50-year anniversary of ESPOC, and I want to thank the chimborazo itself. Thank you.
Agradecemos al doctor Chris de Zeeuw y empezamos en este momento con la ronda de preguntas. Mi micrófono, por favor, activar el micrófono inalámbrico.
Well, my question is that how the results of the app here in Latin America or Ecuador how the output of this app is showing results compared like to America or Europe?
Yes, that's a very good question. So again, all these data have not been published yet, but we see some very interesting data. I would say in general what we see is that, for example, when you look at pre-pulse inhibition, right? This is not the only thing we're doing, we're also doing normal conditioning, classical conditioning, trace conditioning, etc. But if you look at PPI, for example, it seems like that when you're living in a busy city, like we most of us do, and I even call, Riobamba a big city, we have a strong tendency to suppress all sorts of filters. While I'm talking here, I hear these rumors, people talking there, I get light and I get all these impressions. He's maybe a patient waiting for me, I don't know. But there's all sorts of things happening to me, and I'm constantly trying to sort of diminish the input, right? I constantly try to select what's now most important. Okay, your question is most important, so I try to focus on that. But it means that the inhibition of impulses and reflexes is very, very important in our Western-like society. Now, if you look at people, for example, in Africa, we are now reporting tribes in Africa who have never ever seen a car, right? So they are really down to the basics. They don't show any pre-pulse inhibition at all. So they need to hear all the little sounds from birds that can predict that there might be a lion coming, I don't know. I mean, but they are very sensitive, and they stay sensitive, and they don't want to filter out anything of their sensory inputs. So there is indeed very important regional differences, and I'm sure when we have all the data from Riobamba and the Andes Mountains, when we've analyzed it, I'm sure we will see also comparisons between, let's say, people who live really here in Riobamba or who will live really in the mountain area itself. But that's all I can say so far. But it's a very adequate question because we see that already in the data we have that this is taking place. And of course, in the patient group, you need to, of course, always compare it to your local controls.
Muchas gracias por la respuesta. La siguiente pregunta.
Well, good morning, Dr. Chris Zeeuw. Well, this is my questions. I have two questions for you. Is the Blink Lab available for Android phone or just iPhone?
Yes, so we currently developed it for iPhone, right? So we are actually, like today when I'm going to meet Valeria, I will give her a couple of extra iPhones from Blink Lab. We are constantly bringing iPhones to the Blink Lab people here in Riobamba, and that's our basic setup. And it's important because all the circumstances, like the light background, the noise levels, all of that has to be adjusted with AI programs in the same software. And this is what we're doing, but in principle it will also be very easy to modify it for Androids. And so we are expanding it also within the next year. So we hope to get FDA approval next year in the United States and then also for South America. And then at the same time we will have it for all the smartphones that can be used.
So, the app in the 2023
Yes,
available.
yes, it's free. So now that it's true. So you wonder what is then the business model? So, yeah, right?
Right.
Okay, so that's a good question. We're trying to figure that out as well. But there's like three main sources of income. First, there's still the academic institutes, because we show already that the app that we have now is actually working much better than the much more expensive and much more complicated setups that we also developed ourselves in our own institute, and that we also sold to other institutes actually. But it means that those academic institutes, they really want to use the apps, and they really want to pay for this, because you have to understand that all the data are coming in our platform. So although all the applicants will get the data, we will get the data and we will analyze it against the background of all the other controls that we have. So that information, that data set is worth really a lot of money for the academic institutes, and they don't need to buy expensive equipment anymore. But there's two other sources, right? So this is just one. The other one, the second one, is in the clinic. So once we have the FDA and the MEA approval in Europe For clinicians, the same thing will hold. They don't have to do very expensive questionnaires or whatever. They will get quantitative data on a very short notice through these iPhone setups in the clinic and with an analysis report from us, which by then is FDA and MEA approved, we can provide it. So that's definitely worth something. And the third group, to my own surprise, according to marketing analysts, is actually the biggest problem. And that's the third group is actually you, it's all of us. So they think that everybody who can download the app, and let's say you only have to pay maybe 1 cent to download it, and just make it overnight, but that actually many people might be interested in examining their own mental state. See, okay, well, my P-pulse inhibition today is terrible because I was drunk last night, or, you know, for whatever reason, then they expect that many people are interested in getting this information for their own private life. I still have to see that, but again, according to the analyst, this is actually the biggest market. I hope that answers your question.
Yes, and the second, please. Could all this information be reckoned with Alzheimer's time? It is a neurological disease.
Gracias. La última pregunta, por favor, para dar oportunidad a otros. Gracias.
Yeah, so for Alzheimer, of course we are also testing on Alzheimer patients, but I would like to say this, that, which was also a surprise. So, like 10 years ago, we started to, so 10 years ago we started to look at the kindred of people with familial Alzheimer in Colombia, right? So in Colombia you have this group of patients who suffer from a particular mutation in presenilin, and we looked at cerebellum tests in those patients. So we went to Colombia, we did the same test in the mouse models with the same mutation, and surprisingly we found there that the cerebellum symptoms, even though there were no plaques entanglements yet in the cerebellum, which is minor anyhow, but way before they were there, we could detect deficits in motor disorders. So we published this in 2013 in Journal of Clinical Investigation with the Colombians there. And so it wouldn't surprise me, given that, you know, hypercognition, we know very well what's going on in the cerebellum, that we will be able, for sure in the familiar form of Alzheimer, or probably also in the other forms, to see in a very early stage clinical symptoms. So that's all I can say so far.
Muchas gracias por la respuesta. Por favor, tenemos aquí dos compañeros traductores. Si algún joven desea preguntar, por favor, y necesita, o cualquier persona, tenemos dos compañeros, recuerden, para hacer la traducción simultánea. ¿Alguien más que desea preguntar? Allá arriba. Gracias.
Okay, good morning everyone, and doctor. I just want to ask you a question, and I just want to say, how long did this research take place and what motivated you to do it?
Read the question. Yeah, so you mean the Blink Lab investigation. So your question, if I can repeat, is correct. So the question is, how long did it take to develop the Blink Lab, and what was the motivation to develop Blink Lab? Is that correct?
Yes.
Yeah, so again, as I said, we have been thinking about it actually for a while, even before Henk-Jan went to Princeton. So Bas and I and Henk-Jan had been talking about it, but it wasn't really until COVID occurred and that the lab for animal research had to be closed at Princeton, and Henk-Jan was there anyhow. So he felt, with a lot of AI, deep learning people, they're present in Princeton, this was the right moment actually to use computer vision because this is what you need to do, right? If you have your app and you record the eyelid movement and the whole facial movement, you need to have very good computer vision programs, AI programs, to analyze it very quickly and adequately. And I must also say, like, in a much earlier stage, when we already had the basic idea, that quality of computer vision was not there yet. So that came together with COVID, and then once it got going, this was the beginning of 2020, then it went all very, very rapidly, because it was then judged by all the scientists who are looking at valorization patterns, potential patents at Princeton, and this one was ranked #1. So we got an accelerator award, as they call it, so we got extra money, and then there were also already investors who wanted to do the first investment, and then it went, yeah, we are still expanding, so then it went very quickly, and I hope we'll keep going this way.
Muchas gracias. La última pregunta.
Good morning, everyone. I would like to know if you have observed or studied with research about feeding and if it somehow alters the modulation of the up and down of the spikes.
Yes, of course, yeah. So we cannot, unfortunately, directly test the down-bound and up-bound activity in the clinic with normal electrophysiological recordings. We can do some recordings in patients with epilepsy, but I should say that we have developed a new technique to investigate activity with ultrasound. And so particularly when you have patients which skull is different, for example during an oncological operation, so when the tumor is removed, we actually can test the activity of the brain with ultrasound. And there we are trying to also visualize the zones, and we have some first indications that it might work, but it's still very much in the beginning stage. Now, so this is one aspect. Of course, we do the same thing with fMRI, but that is still difficult because the temporal resolution and the spatial resolution is limited. Now, you may have seen a paper in Science that was published 2 weeks ago, where they actually showed in rats that the temporal resolution with fMRI can be improved dramatically, not by looking at the Bolt signal, but something that's done before that, a very high temporal resolution. So if that will be available for humans, then I think actually, and this will probably take a few years, then we should be able to also see up-bound and down-bound activity in humans with non-invasive methods. And we know it's there because we have been doing recordings in non-human primates, and they see the same up and down activity in the primates. So, but I don't think anyone ever showed it very precisely in humans yet.
Muchísimas gracias por la respuesta. Vamos a dar por concluida la conferencia magistral del Dr. de Zeeuw, y le agradecemos mucho a nombre de la Escuela Superior Politécnica de Chimborazo y de la Red Ecuatoriana de Universidades y Escuelas Politécnicas para la Investigación y el Posgrado REDU. Muchísimas gracias.
Okay guys, we are very grateful with the participation of Dr. Everybody, please, another clap.